Dear student, in genetics part 3 we'll be talking about deviation from Mendelism. So Mendel's 3 laws were truly applicable to pea plant and several other plants and animals also, but in wide range the Mendel's law seems to be deviated from the original law. Let's look at 3 laws: dominant, segregation and independent assortment. Out of these 3, segregation is the universal law. Because universal law, it is because you can see the gamete always segregate during the meiosis in the sexually reproducing organism, and again the paired condition is restored during the fertilization. Now if you look at the law of dominance, the deviation is observed in 2 different forms. One is incomplete dominance and codominance. Now incomplete dominance, the 2 alleles in heterozygous condition may produce an intermediate phenotype when both are present rather than one fully determining the phenotype. Codominance, 2 alleles in heterozygous condition will simultaneously expressed when both are present rather than one fully determining the phenotype. Now what does it indicate? In law of dominance, according to the Mendel, in F1 the one which is dominant is expressed, whereas if you look at the incomplete dominance, here both in F1 both the alleles which are present, they will partially express their product, whereas in case of codominance both the allele will fully express their product. Okay, so product means their character. Now independent assortment, deviation, here we have 2 things in the syllabus: multiple alleles and pleiotropy. So multiple alleles means rather than 2 there exists multiple allele, that is one character is influenced by number of genes. Pleiotropy, one gene may control more than one character. So all these possibilities are there. Now here I have taken one imaginary example to explain the difference between the incomplete dominance and codominance. So here considering the character that I have chosen to study is the color of flower. There are 2 different traits. R is dominant, capital R is red is dominant trait, whereas white is recessive trait. So we start with the pure lines. Here the gametes are produced, and if you look at the F1 hybrid, then both are partially expressing their both the alleles are partially expressing their character. Okay, so or trait. Okay, that means now there is no red color that will appear in F1, but instead of red, pink will appear. And if you look at the F2, if you do the F2 cross, F1 into F1 for F2 cross, then you will see the ratio is 1:2:1. So 1 red, 2 pink and 1 white. And this is again deviation from normal 3:1 ratio that we have seen in the law of dominance in the Mendelism. Okay. Again, if you look at the codominance, similarly parents are there, then gametes will form, meiosis will occur, and again in F1 both these alleles, capital R and small r, they will express their character. So what you will see in the phenotype, you will see patches of pink, you will see patches of red and white flower. Okay, and if you look at the F2 ratio, it will be 1:2:1, 1 red, 2 patchy and 1 white. So this is the difference which you can clearly. Now there is another example to study the codominance is the blood group. You know ABO blood group system in the human, and let's briefly discuss. Let's assume that there are genes which are present on the chromosome, and they are these genes are expressing certain proteins which are called antigen, which are expressed on the surface of RBC. So if the gene expresses A type of antigen, we say blood group is A. when it expresses B type of antigen, we say blood group is B. And if it does not express any kind of antigen, we say the person is having blood group O. And if it expresses both A and B type of antigen, then we say the blood group is AB. So now if you look at the genotype, blood group A can have both the alleles for A or one allele for A, and IO means recessive allele, means it is not expressing the. So once IA is dominant, so if IA and IO are there, then A blood group will be there. So the person who is having blood group A will have two kind of you can say genotype, either this or this. Okay. Similarly, B blood group, the alleles will be both will be homozygous or maybe in the heterozygous condition. AB here you can see this is typical example of codominance, because one of the allele is A which expresses A type of protein, another allele is which expresses B type of protein. So here you will see both are expressing on the surface, hence the person's blood group is AB. O blood group means they are not expressing any protein. So in the above example, you can also see that more than two alleles are controlling one character. Okay, there are three alleles you can see, so more than two. So such alleles are called multiple alleles. Now all these alleles occupy the same specific locus on the chromosome. In the diploid cell, only two of these could be present. Either any of these two, three could never be present in the diploid cell. Okay, but they exist in the gene pool at the time on the homologous chromosome. So let's discuss one example. Suppose the male parent is with blood group A and female is with blood group, and they are their genotypes. Now during the meiosis gamete formation will occur, and then you will see in the offspring what all possibilities are there or probability of blood groups are there. So if you look at the genotype, on the basis of that you can make out the phenotype. Offspring may have AB blood group, so 50% offspring are with AB blood group and 50% offspring are with B blood group. Okay, where the parents are having A and B blood group. Can you see? Okay. Now next is polygenic inheritance. Now one particular trait is determined by more than one gene. All such genes are non-allelic. Last time what blood group we were talking about, where one character is controlled by more than two alleles, that is multiple gene, multiple you can say alleles. Whereas here we are talking about genes, not alleles. So more than one gene controlling a particular character. Individually, each gene has little effect on the phenotype, but their combined effect is very significant. Let's discuss this example and see this under the polygenic inheritance. We can study the example of skin color in human, which is controlled by melanin synthesis. Now it is a pigment. The synthesis of melanin is controlled by three different genes. Let's assume A, B and C, and they're located on different chromosomes. Okay, so the person with dominant A, B, C, okay, will be the darkest, and with the recessive alleles A, B, C, it will be white, or we say albino. The skin pigmentation is determined by number of dominant gene present. Each dominant gene is responsible for synthesis of a fixed amount of melanin. What does that mean? If suppose more of the dominant gene is there, suppose A, B, B, C and small c, and if A, B, B, small c, so this is having one extra dominant. See, here are 5 dominant you can say alleles. Here you can say only 4 dominant alleles. Okay, so this will be more darker than this. So more are the number of dominant alleles, more will be the darker will be the color. Okay, So, and this is called quantitative expression. Since the effect of gene is additive, the amount of melanin produced is always proportional to the number of dominant gene. Okay, additive means if 1 is there, 2 is there, and if 3 more dark is there. In this way, interaction of 2 or more genes which are non-allelic give rise to quantitative inheritance, as the amount of color produced will depend on the number of dominant gene. There are two kind of inheritance. One is quantitative. The polygenic inheritance is the example of quantitative inheritance. In qualitative inheritance, there is no intermediate, like we have color of the flower, maybe white or purple, like that. Now, this phenomena is also responsible for continuous variation. We will discuss this point in chapter evolution. Let's discuss pleiotropy or pleiotropism. A responsibility of a gene for more than one phenotypic effect, often seemingly unrelated, is known as pleiotropy. Let's discuss this with the example of phenylketonuria. So phenylketonuria is a genetic disorder and it is caused due to the accumulation of phenylalanine. Phenylalanine is the amino acid which is aromatic amino acid. And what is the genetic defect? The genetic defect is in the PAH gene. Now this gene actually codes for the enzyme phenylalanine hydroxylase. Phenylalanine hydroxylase, what it do? In the body, it catalyzes the conversion of phenylalanine to tyrosine. Tyrosine is another amino acid. So if this enzyme is defective or the gene for this enzyme is defective, then two things happen. One is the accumulation of phenylalanine in the body, in the cells, blood, and the deficiency of tyrosine. Okay. So phenylalanine, when accumulated, it gets excreted in urine, and this is called phenylketonuria. And if you look at this person suffering from phenylketonuria, then you'll see different types of symptoms, but they are unrelated, like mental retardation because of the accumulation of phenylalanine in the brain. Then as the tyrosine is required for melanin production, therefore symptoms of reduced coloration of hair, skin are observed, and it also responsible for eczema of skin. So different phenotype. So one gene is affecting the different phenotype, hence we say it is pleiotropism. Another example of pleiotropism is sickle cell anemia. Now sickle cell anemia is again a genetic disorder in which the abnormal you can say hemoglobin is produced. As a result, the shape of the RBC will become sickle in shape, and these sickle shape RBC they are not very efficient in carrying the oxygen. Moreover, when they pass through the blood capillaries, they get stuck in the blood capillaries and causes lysis of blood cells, and this all results in anemia. Okay. Now the defect is mainly in the beta globin gene of hemoglobin, and once this defect is there, again multiple symptoms are produced. All these symptoms are related to circulatory and respiratory system, along with the pain attacks, swelling in hands and feet. So one gene is causing multiple phenotypic effect. So again, this is the example of pleiotropism. Now sickle cell anemia is also example of codominance. It is because the person who is carrier for sickle cell anemia is the gene is present in the heterozygous condition. One is normal gene, another one is the defective gene, and the person who is homozygous for sickle cell anemia, where both the alleles are defective, that person will not survive much. Okay, so if you look at, this is the normal hemoglobin gene. A represent the normal, as defective, and if the heterozygous condition, one normal and one sickle cell anemia gene. and here 50% cells are normal, so the person will survive, and it is known as carrier. Whereas if you see homozygous condition, all the cells will carry the defective gene for hemoglobin, and the person will not survive. And this disorder, since this get expressed fully, or you can say this get expressed in the recessive condition, and the defect is on the autosome, so this is also known as autosomal recessive disorder. So sickle cell anemia is the example of autosomal disorder, recessive disorder. Now there is another thing that we would like to talk about sickle cell anemia, that the carrier of sickle cell anemia are highly resistant to malaria. The sickle cell RBC will not express certain protein that help in binding of pathogen or parasite to RBC. Moreover, the sickle cell release some chemical that makes the environment unfavorable for the growth of parasite. Hence, malarial parasite could not grow in the person who is suffering from sickle cell anemia. Now this is the example of natural selection, or you can say evolution by natural selection, because here the sickle cell anemia population is selected over malaria suffering population. Okay, so very common in the place where the malaria is endemic. So again, this is the example of evolution. This is all. Will continue rest of the things in next part of genetics. Thank you.